Welcome to another episode from the Veterinary Research Channel. Today, we delve into a fascinating study published in the Journal of Veterinary Internal Medicine in 2023, exploring the genetic underpinnings of a concerning neurological disorder in horses known as equine neuroaxonal dystrophy, often abbreviated as eNAD, or equine degenerative myeloencephalopathy, also known as EDM. This research, conducted by Yunjua Ma, Sikongpum, Callum G. Donnelly, Sharmila Ghosh, Andrew D. Miller, Kevin Woolard, and Carrie J. Finno, sought to understand if variations in genes responsible for the transport of vitamin E could contribute to an increased risk of this debilitating condition in quarter horses. Equine neuroaxonal dystrophy, or equine degenerative myeloencephalopathy, is recognized as an inherited neurodegenerative disease. It is believed to be connected to a deficiency of vitamin E, particularly during the crucial first year of a horse's life. The clinical signs of this condition are quite characteristic, often presenting as a symmetrical lack of coordination, which veterinarians grade on a scale where a higher grade indicates more severe issues. Horses might exhibit a wide-based stance even when resting, show difficulties with their sense of body position, known as proprioceptive deficits, and typically have lower levels of vitamin E, specifically alpha-tocopherol, in their blood serum. Interestingly, even after a horse passes its first year, the concentrations of this crucial form of vitamin E in their blood tend to remain lower compared to healthy horses of the same age. The good news is that equine neuroaxonal dystrophy, or equine degenerative myeloencephalopathy, can potentially be prevented in foals that are genetically predisposed to the condition. This preventative measure involves supplementing the mares with high doses of a water-soluble form of alpha-tocopherol, specifically RR or alpha-tocopherol, during the final 3 months of their pregnancy. This supplementation is then continued in the foals themselves throughout their first 2 years of life. Currently, the only definitive way to diagnose equine neuroaxonal dystrophy, or equine degenerative myeloencephalopathy, is through a post-mortem examination. This involves a detailed histological evaluation of the brainstem and spinal cord after the horse has passed away. While scientists have recently developed a biomarker test that looks for phosphorylated neurofilament heavy chain in the blood serum, which has shown some promise in diagnosing equine neuroaxonal dystrophy, or equine degenerative myeloencephalopathy, its overall reliability is still considered low. In the realm of human health, there is a comparable inherited disease called ataxia with vitamin E deficiency, often referred to as AVED. This condition is caused by harmful variations in the gene responsible for the alpha-tocopherol transfer protein, known as TTPA. This protein plays a major role in moving alpha-tocopherol into the lipoproteins that are secreted by the liver and circulate in the plasma. Interestingly, research has shown that equine neuroaxonal dystrophy, or equine degenerative myeloencephalopathy in horses, is not associated with genetic mutations in this alpha-tocopherol transfer protein gene. However, the researchers in this study hypothesized that variations in other genes involved in how vitamin E is taken up by the body, distributed to different tissues, and metabolized, could potentially influence a horse's risk of developing equine neuroaxonal dystrophy, or equine degenerative myeloencephalopathy. Previous studies have indicated that the absorption of vitamin E and its transport to target tissues do not seem to be altered in horses with equine neuroaxonal dystrophy, or equine degenerative myeloencephalopathy. However, there is evidence suggesting that the metabolism of vitamin E is increased in quarter horses affected by this condition. Furthermore, a prior study found greater production of an enzyme in the liver called CYP4F2, which is the primary enzyme responsible for breaking down vitamin E, in horses with equine neuroaxonal dystrophy, or equine degenerative myeloencephalopathy, compared to unaffected horses. Given these findings, while the researchers chose to investigate all known candidate genes involved in vitamin E pathways, their main hypothesis was that genetic variations in genes involved in vitamin E metabolism, particularly CYP4F2, would be linked to a higher risk of equine neuroaxonal dystrophy, or equine degenerative myeloencephalopathy. The study embarked on a comprehensive genetic analysis, starting with whole genome sequencing of a group of horses. This initial group included 9 quarter horses confirmed to have equine neuroaxonal dystrophy, or equine degenerative myeloencephalopathy, through post-mortem examination, and 32 control quarter horses that were not affected. Following this initial screening, a larger validation study was conducted involving 39 quarter horses diagnosed with equine neuroaxonal dystrophy, or equine degenerative myeloencephalopathy, of which 23 had their diagnosis confirmed post-mortem. This validation group was compared to 68 control quarter horses, with 7 of these having been confirmed as unaffected via post-mortem examination. Additionally, The researchers utilized publicly available genetic data from a diverse group of 504 horses across 47 different breeds to further investigate the prevalence of any identified genetic variations. The research team meticulously examined 28 candidate genes known to be involved in vitamin E absorption, transport, and metabolism. They identified genetic variants within these genes using advanced genomic techniques. After rigorous filtering and statistical analysis, the initial whole genome sequencing data revealed 43 variants in 15 of these vitamin E candidate genes that showed a significant association with equine neuroaxonal dystrophy or equine degenerative myeloencephalopathy. However, further scrutiny using a tool called Integrative Genome Viewer indicated that 13 of these initially identified variants were likely incorrect classifications due to repetitive sequences in the genome. This left 30 variants that were then taken forward for further investigation in the validation cohort of horses. In the validation study, the researchers focused on these 30 remaining genetic variants using a method called mass array genotyping. After further quality control checks, 21 variants were excluded due to low minor allele frequencies, and one failed the genotyping process itself. This left 9 variants that were subjected to case control association testing. To account for the fact that they were testing multiple variants, a statistical correction called Bonferroni correction was applied, setting a new threshold for significance. The results of this validation phase were particularly interesting. 2 specific variations, known as single nucleotide polymorphisms or SNPs, within the CD36 gene showed a significant association with equine neuroaxonal dystrophy or equine degenerative myeloencephalopathy. These 2 variations were located at specific positions on chromosome 4 of the equine genome, specifically at position 726485 and 731082. This significant association was observed both when considering all clinically diagnosed cases of equine neuroaxonal dystrophy or equine degenerative myeloencephalopathy together, and when focusing only on the cases that were confirmed through post-mortem examination. Despite this statistically significant link, the researchers noted that the frequency of these variant forms of the CD36 gene was relatively low even in the post-mortem confirmed cases of equine neuroaxonal dystrophy or equine degenerative myeloencephalopathy, ranging from 0.22 to 0.26. When they looked at the publicly available genetic data from a broader range of horses, these variant frequencies ranged from 0.06 to 0.1. Further analysis of these 2 CD36 variations indicated that they are located within the introns of the gene. Introns are non-coding regions within a gene. For the variation at position 731,082 on chromosome 4, it is predicted to be located within the introns of 6 different versions of the CD36 gene and has a genomic evolutionary rate profiling score of 0.29. The genomic evolutionary rate profiling score is an indicator of how conserved a particular genetic region is across evolution. A higher score suggests greater conservation and potentially greater functional importance. For the other significant variation at position 726,485 on chromosome 4, it is also predicted to be within the introns of 6 CD36 gene versions and has a genomic evolutionary rate profiling score of 1.00. Notably, minor allele frequency scores for these specific single nucleotide polymorphisms were not available in the Ensemble database, a comprehensive resource for genomic information. While the association with these 2 intronic CD36 single nucleotide polymorphisms was statistically significant, the researchers made a crucial observation. A considerable number of the post-mortem confirmed equine neuroaxonal dystrophy or equine degenerative myeloencephalopathy cases did not carry the alternate form of these genetic variations. Specifically, for the variation at position 726485, 12 out of the 23 post-mortem confirmed cases were wild-type, meaning they had the more common version of the gene. Similarly, for the variation at position 731082, 10 out of the 23 post-mortem confirmed cases were also wild-type. Interestingly, when the analysis was restricted to only the clinically diagnosed cases of equine neuroaxonal dystrophy or equine degenerative myeloencephalopathy, meaning those diagnosed based on clinical signs and not necessarily confirmed postmortem, a third single nucleotide polymorphism at position 39083023 on chromosome 26 also showed a significant association with the condition. This variation is located within an intron of the ABCG1 gene. The ABCG1 gene is predicted to have 4 different versions, and this single nucleotide polymorphism resides within the introns of all of them. It has a genomic evolutionary rate profiling score of -1.27, and its highest minor allele frequency reported in the Ensemble database is 0.83. To further understand the prevalence of these top 3 single nucleotide polymorphisms, the researchers examined the publicly available genetic data from 504 horses across 47 breeds. The overall frequencies of these variant forms ranged from 0.06 to 0.1 across all these horses. When they looked at the frequencies within specific breeds that had at least 25 individual horses sampled, they found a wider range. For the single nucleotide polymorphism at position 731,082 on chromosome 4, the within-breed frequencies varied from 0.03 in franchise montane horses to 0.30 in warm-blood horses. For the variation at position 726,485 on chromosome 4, the frequencies ranged from 0.02 in Shetland and standard-bred horses to 0.23 in warmbloods. Finally, for the single nucleotide polymorphism in the ABCG1 gene on chromosome 26, the frequencies ranged from 0.01 in standardbreds to 0.3 in franchise montane horses. The researchers initiated their discussion by reiterating the understanding that in quarter horses, the development of equine neuroaxonal dystrophy or equine degenerative myeloencephalopathy requires both a vitamin E deficiency after birth and a genetic predisposition. Prior research had identified abnormalities in vitamin E metabolism in quarter horses with this condition, primarily characterized by an increased breakdown of alpha-tocopherol in both their blood serum and urine following the administration of an oral dose of RR alpha-tocopherol. That same earlier study also found higher levels of the CYP4F2 enzyme in the liver, the main enzyme responsible for vitamin E metabolism, in affected horses compared to unaffected ones. Therefore, the current study aimed to investigate genetic variations in genes involved in the uptake, transport, and metabolism of vitamin E, including CYP4F2 itself. While this study did not identify any significant genetic variations in the CYP4F2 gene, it did find a significant association between 2 intronic single nucleotide polymorphisms in the CD36 gene and equine neuroaxonal dystrophy or equine degenerative myeloencephalopathy in quarter horses in both the clinically diagnosed and post-mortem confirmed groups. Additionally, a third single nucleotide polymorphism within the ABCG1 gene on chromosome 26 was significantly associated with the condition, but only in the clinically diagnosed group. The CD36 gene, also known as cluster determinant 36, plays a vital role in the uptake of fatty acids in numerous tissues. This transporter belongs to the scavenger receptor family and is involved in taking up oxidized low-density lipoproteins from the bloodstream. Interestingly, alpha-tocopherol has been shown to reduce the production of CD36, both in laboratory studies and in living organisms. In humans, a specific intronic single nucleotide polymorphism in the CD36 gene has been linked to plasma alpha-tocopherol concentrations, suggesting that CD36 might be involved in either the absorption of vitamin E in the intestines or its uptake by tissues. Thus, several lines of evidence point to a role for CD36 in the uptake of vitamin E, either directly or indirectly. However, given that the intestinal absorption of vitamin E in horses with equine neuroaxonal dystrophy or equine degenerative myeloencephalopathy is comparable to unaffected horses, and the uptake of vitamin E into the liver and central nervous system is also similar after vitamin E supplementation, the researchers suggest that variations in CD36 are unlikely to be the primary cause of equine neuroaxonal dystrophy or equine degenerative myeloencephalopathy. Furthermore, the 2 CD36 associated single nucleotide polymorphisms identified in this study are located in non-coding intronic regions and have genomic evolutionary rate profiling scores below 2, which typically does not indicate a high level of evolutionary conservation. However, the researchers acknowledge that this method might have limitations in identifying harmful mutations, particularly in non-coding regions, as regulatory elements are not always highly conserved in their sequence. Despite the strong statistical association found between equine neuroaxonal dystrophy or equine degenerative myeloencephalopathy and these 2 intronic CD36 single nucleotide polymorphisms, the significant finding that many post-mortem confirmed cases did not possess the alternate version of these genes and the relatively high frequency of these variants in other breeds raises concerns about the true nature of this association. For instance, thoroughbred horses are known to be less susceptible to equine neuroaxonal dystrophy or equine degenerative myeloencephalopathy, yet the frequencies of these CD36 variants on chromosome 4 were found to be relatively high in this breed, ranging from 0.15 to 0.18. Detailed examination of the genetic makeup of the horses in the validation study revealed that none of the horses in that group were homozygous for the less common version of either of these CD36 single nucleotide polymorphisms. In the larger public dataset of 504 horses, only 3 horses were homozygous for the less common version of each of these single nucleotide polymorphisms. The researchers suggest that the lower frequency of these less common versions in quarter horses might have led to a false association in the study, especially considering that the frequency in the public database of quarter horse samples was only between 0.06 and 0.08. They also point out that their study population of quarter horses might have inadvertently included a subpopulation with a higher frequency of these genetic variants, and while they attempted to match cases and controls by subpopulation, this potential stratification could have led to false positive results. This highlights the critical importance of evaluating the frequencies of genetic variants across different breeds after identifying potential associations in order to avoid drawing incorrect conclusions. The third single nucleotide polymorphism on chromosome 26, which was found to be significantly associated with equine neuroaxonal dystrophy or equine degenerative myeloencephalopathy only in the clinically diagnosed group, is located within the ABCG1 gene. ABCG1 encodes for an ATP-binding cassette protein involved in transporting various molecules across cell membranes. This protein has been implicated in the efflux of vitamin E from cells, and vitamin E metabolism has been shown to be abnormal in mice lacking the ABG1 gene. However, due to the limited association of this ABCG1 single nucleotide polymorphism only with the clinically diagnosed cases and its high frequency in many other breeds, the researchers believe that this variant is likely not a strong candidate for increasing the risk of developing equine neuroaxonal dystrophy or equine degenerative myeloencephalopathy. The researchers also acknowledged a limitation of their study, which was the preselection of candidate genes based on existing knowledge. While they aim to include as many documented vitamin E-related genes as possible, they recognize that other genes potentially involved in the development of equine neuroaxonal dystrophy or equine degenerative myeloencephalopathy might have been overlooked. Their selection was primarily based on a comprehensive review of human literature. Furthermore, despite using both computational algorithms and visual inspection of sequencing data to identify larger structural variations in the genome, they note that errors in the reference genome assembly could have led to missing larger rearrangements within the candidate genes that might be associated with the disease. They suggest that future long-range sequencing efforts are necessary to better assemble the genomes of affected and unaffected horses to identify such potential rearrangements. Despite the lack of a strong association with any specific candidate variant in this particular study, the researchers emphasize that a genetic component to equine neuroaxonal dystrophy or equine degenerative myeloencephalopathy remains strongly supported by the occurrence of the disease within families and large-scale investigations on breeding farms. In conclusion, this study identified a significant association between 2 intronic single nucleotide polymorphisms in the CD36 gene and equine neuroaxonal dystrophy, or equine degenerative myeloencephalopathy, in quarter horses. However, the fact that many post-mortem confirmed cases of equine neuroaxonal dystrophy or equine degenerative myeloencephalopathy were wild-type for these variants suggests that this might be a false positive association, or that there could be genetic heterogeneity for equine neuroaxonal dystrophy or equine degenerative myeloencephalopathy within the quarter horse breed. Thank you for listening to the Veterinary Research Channel. A link for the referenced paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.